Hi, I'm Sarah Kuebbing. I'm an assistant professor in the Department of Biological Sciences at the University of Pittsburgh. In our lab group, we really like to go outside, and we go outside all times of the year, which you'll see a lot through this video since we made it in the winter. But we go out in the field and we observe plants, we collect soil samples, we take those back to the lab where we process those soil samples. We sometimes even go into museums where we use herbarium samples to answer questions about how plants in places like these forests are responding to invasions.
Hi, my name is Jess and I love plants. Luckily for me, I work in a plant ecology lab. So the Kuebbing Lab, led by Dr. Sarah Kuebbing, who you just heard from, wants to examine a lot of different aspects about plants and the forest they live in. So we're looking into plant phenology, which is when plants produce their leaves or their seeds or their flowers, and how that is affected by climate change. We're looking into plant invasion, so non-native plants going into forests around here, and are they affecting the native plants? And then we're also thinking about land use history. So when the land was a mine or a farm and then we turn it into a forest, how does it affect the way that the forest and the plants behave? A science lab is made up of a lot of different kinds of researchers who all want to answer these questions. So we have undergraduate researchers. They are still in college and they work in the lab part-time while they're still going to classes. We have PhD students who are doing a 5 or sometimes 6-year program to really explore their personal questions about plant ecology. And then we have postdocs, and these students have finished their PhDs and they're doing shorter 2-year research projects to look further into a plant subject. Last, we have my job, the lab manager. I think my job is the best because I get to work on all of the projects in the Kuebbing Lab. So come with me and I'll show you just some of the questions we are looking into right now. Let's start our Kuebbing Lab journey in the forest. Here, Naeem and Ellen are collecting branches of the dogwood tree. We collected many different branches and put them in our growth chambers with different amounts of light to see how this light changes when they leaf out. Here you can see they just have small little green buds, and then after a week or so, you can see that they're really leafing out. Let's go back into the field and take a look at Rachel's project. Rachel and Ellen are collecting some plants in the snow. It's cold, but the plants are still there, waiting for spring. Rachel is creating a competition experiment between 3 species of plants in the greenhouse. These are all invasive plant species. Invasive plants are plants that originated from another place, and now they can do better and sometimes take over the places where native plants are living. You can see how Rachel's plants have gone from little tiny seedlings in the snow to big flourishing pots of green in just a couple weeks.
Hi there, my name is Rachel. I'm a 3rd-year graduate student in Sarah's lab, and I study how invasive plant species compete with one another. And so in this project in particular, I am studying how these different invasive forest plants are competing with each other, and specifically looking at how the timing of their life cycle events are affecting their competition.
Let's go back into the lab and see what Ellen is up to.
Hi, my name's Ellen. I'm a 3rd-year undergrad in Sarah's lab. My major is in ecology and evolution, so I love plants. I have a bunch of house plants at home, which I love. Behind me, I'm doing soil texture analysis, which is when we put soil and a bunch of other stuff in these graduated cylinders to see how much of the soil is made of sand, silt, or clay. And I'm doing this because I am doing a tree-based microhabitat project where I'm looking at what factors may facilitate invasion. So I went out to the woods and counted all the invasive species around different trees, and then I took samples of the soil. So like factors that might facilitate invasions are like the type of tree that the plants are growing around, or the soil texture, the soil pH, or like fungal interactions within the soil.
Really exciting stuff, Ellen. Let's stay in the lab and see what Naeem is up to.
Hi, my name is Naeem Aziz. I am a senior ecology and evolution major, and my research interests include the spread of invasive plants and the timing of different plant life cycle stages.
All right, let's let Naeem get back to his measuring, and we'll check in with Amoy and their projects on invasive knotweed.
Hi, my name is Amoy, and I'm currently in the Kuebbing Lab. I study invasive plants, and I have a project running right now that takes a look at the invasive shrub, knotweed. I'm interested in Japanese knotweed, giant knotweed, and the byproduct of that, which is the Bohemian knotweed, otherwise known as hybrid knotweed. And as a hybrid of the two parents, Japanese and giant, I find it interesting because it is actually a better invader, and I want to know what traits allow it to do that. And in order to understand it, I have to understand what environments it's surviving in. And so I've been running some tests on a variety of things. In particular, in my hand right now, I have some soil samples that I've been analyzing. Some tests that I've been running look at the pH, whether or not the soil is acidic, basic, or neutral. What's the texture of the soil, and what's the overall ability of the soil to withhold moisture or to keep moisture. There's still a lot to be learned about invasive plants, and everything we learn can help us better manage them.
Awesome, thank you, Amoy. Let's go back out into the forest with Marion and see what she's up to.
Hi, I'm Marion Holmes, and I'm a postdoctoral researcher in the Kuebbing Lab. My main research interest is understanding how forests that we go and hang out in today are different from those forests that would have preceded European colonization. A lot of the forests that you might know and love, like North Park or even Frick Park or Raccoon Creek State Park, if you were to go back 100 years ago and get in your time machine, you wouldn't see forests at all. You'd probably see fields used for agriculture, maybe some freshly logged areas. A lot of the forests that we have today is actually quite young in comparison and regrew after those lands were abandoned 80 to 40 years ago. The forests are therefore different from what would have been there before clearance, and some of those differences include non-native species. There are a lot of non-native species in young forests, and I want to understand why they're there, what that tells us about how forests function, and also what that can tell us about how to manage these species if they're bad for native ecosystems.
Awesome, thank you for explaining all that, Marion. And while Marion and I were out in the field, she showed me some really cool eastern skunk cabbage. These flowers actually produce their own heat and melt the snow around them, and they create a cozy little spot for insects to pollinate them. They also can live up to 20 years. The next time you're in the woods, make sure to keep an eye out for skunk cabbages poking their flowers out of the snow, and see what other really cool plants you can find. Thanks for listening.